as follows. We start with a 10 to 15 minute baseline. We record neural activity, give random rewards, and use 2 selected neurons to calibrate the task difficulty so roughly 30% of trials end in a success or failure by chance. The animal then undergoes a 60-minute testing session, with each trial ending in a success, failure, or timeout, followed by roughly 10 seconds between the trials to allow the animal to obtain a reward if necessary. Finally, you can see here the experimental setup with the animal in the cage, the connected electrodes, and the sugar solution dispenser for providing a reward right here. In order to assess the animal's location in the cage and subsequently its motivation for reward, we recorded videos like this of the animal's position in the cage across all testing sessions. So while we're just beginning data collection, we have successfully set up the neurofeedback task and tested our pilot animals. So today I'm going to share with you our validation results and preliminary findings through a series of 5 questions. The first question is whether we can record single neuron spiking activity in real time. And as you can see here, the answer is yes. We were able to isolate individual neural activity from a number of different channels simultaneously. The second question is whether we can use this neural activity to control the frequency of a sound played to the animal. To do this, we define the cursor value, which is the difference between the smooth spike trains of the up and down units, which again increase and decrease the auditory frequency respectively. We set limits on the rewarded and unrewarded targets, and then map the cursor value to the auditory frequency played using a second order polynomial graphed here on the left. In addition, in order to ensure the cursor values used in real time are the same as the ones used in offline analysis, we confirm that the online cursor values graphed in blue, which were calculated in real time, are the same as the offline cursor values obtained by back calculating the cursor value from the required trains, which we later analyzed, with an R-squared value between the curves of 0.94. Next, we ask whether neural firing rate does increase for the up unit or decrease for the down unit, since the goal is to train animals to increase the cursor value. Using a z-scored firing rate, we saw that in time immediately before success, there's a sharp increase in the up unit firing rate and a slight dip in the down unit firing rate as expected. To better quantify this, we define the modulation depth, which is the average firing rate of a neuron in the 1 second window before success minus the average firing rate in the 1 second window after success. With this metric, we see that the up neuron has a very positive modulation depth that is significantly greater than 0, while the down neuron has a slightly negative modulation depth as expected. The fourth question is whether there are any early signs of learning. Comparing the modulation depth from the first 3 days of testing to the last 3 days of testing on our pilot animal, we see that the modulation depth increases for the up neuron and decreases for the down neuron, indicating that the animal does learn to control its neural activity. We saw further evidence of this in the histogram of auditory frequencies, where the mean frequency increased slightly but significantly between the first 3 days of testing to the last 3 days, with a p-value of less than 0.01. Finally, there was evidence of learning within the sessions over the last 3 days, where the number of events per minute for rewarded targets increased from the first 10 minutes of testing to the last 10 minutes of testing, while the number of events per minute for rewarded targets decreased over the same time period. Lastly, we want to ask whether we could track the animal's location and use it to gain insights into the motivational state of the animal. As seen in this video, we were able to successfully extract the position of various body parts by using the open source DeepLab-Cut software, which uses machine learning to train deep neural networks in order to extract and follow animal location. Using this data, we're able to extract information about the animal's behavior during the task. For example, we compared the mean nose distance from the sucrose solution dispenser following success on the first 3 days versus the last 3 days. With this, we can see the animal was much more motivated for a reward in the last 3 days as compared to the first 3 days, as also can be seen in its mean time to retrieve its reward being much lower in the later days. So in summary, we showed that 1, we can record single unit neural activity in real time and use it to control the frequency of a sound through neurofeedback. 2, we can train animals to control single neurons in the hippocampus. And 3, we can determine animal behavior and motivation by accurately tracking position. For next steps, we are currently testing our first 2 animals. And once we obtain sufficient data on all 10 animals, we will assess how activity in the striatum changes during learning. So why should you care? There are many reasons, but I'll let you know about one application. With a rapidly aging population, dementia and memory loss are increasingly prominent burdens on our global healthcare system, affecting roughly 50 million people worldwide each year, with no current medications or treatments able to fully slow or stop its progression. A novel approach to investigate this is to use deep brain stimulation, where electrodes are implanted into the brain to deliver current to a targeted region. However, this technique has been largely ineffective because memory networks in the brain are very intricate, and this approach, because of its core stimulation, bombards entire memory networks with synchronous stimulation, disrupting these memory circuits. On the other hand, a neurofeedback approach, such as the one demonstrated in our study, offers a potential closed-loop approach to modulate the activity of individual neurons and very precisely control memory networks in the brain without requiring stimulation, to not only improve memory, but also, and more importantly, understand how these memory circuits work. So these are my references, and I would like to thank you all very much for listening and give a special thank you to Creme, Dr. Valiante, and the entire Non2Brain Laboratory for their support, as well as to UNIR for organizing the event and the University of Toronto for sponsoring my summer research, and I'd be happy to take any questions.
Thank you, Michael, for your presentation. Now I would like to address the judges and ask if you have any questions. So judges, you can please unmute yourself and then ask your questions to Michael.
Hi, Michael. That was a great presentation. I really enjoyed it. With regards to your applications with dementia, what would be some of your clinical hurdles to moving into that application and demonstrating the feasibility?
Yeah, for sure. I think the major, I can just go back to that slide, but I think the major application is in when you're treating patients, you mainly have to focus on studying epilepsy patients. So because when you're doing these studies and you're doing them in humans, you do require to have electrodes implanted into the brain. It's often not practical to do this research on, I guess, people who don't already need to have the electrodes implanted. So I guess the limitation is there have been some differences shown between people's brains from patients with epilepsy compared to patients without epilepsy. So I think that's a major clinical hurdle in terms of getting over that. But that said, there are many investigating implanting electrodes into the brain for memory loss. So it is there in the clinical side, but it's just there is that hurdle of actually having to implant those electrodes.
Great, thank you so much.
Hi, Michael. Great presentation. Thank you for that. Very interesting. I just had a follow-up question, I guess, to that. You spoke about the differences between the humans and the rodents, but then what are the similarities that would kind of help your research saying that, you know, what you found in the rodent can be translatable in the human?
Yeah, so I think the thing with like in implementing these technologies is we know that in humans, this hippocampal single neuron learning can occur, but I guess the question is like, how does it occur? And understanding whether the network that we believe facilitates this learning is actually the one that does it. And if we understand the interplay between those two systems, it would help us better understand how we could target certain regions of the brain to help treat memory loss and dementia and stuff like that. So just using the rodent model to just understand those circuits.
Okay, so we are now going to move on to the next presenter. Thank you, Michael, again. Raymond, are you ready to present?
Hi, yes. Okay, thanks everyone for joining today. I'm going to be talking about my project this summer with the Fernandez-Gonzalez lab at U of T, where we've been working to show that adherens junctions and integrins cooperate during Drosophila embryonic wound closure. But they often move as a group, and in order to do so, they need to coordinate and communicate with one another. An important example of this is cancer metastasis, where a group of cells uses collective migration to separate from the primary tumor and spread throughout the body. Understanding the processes that underlie this collective migration is important for cancer therapies that want to target metastasis. Another important example is morphogenesis, which is the series of tissue reorganizations that give tissues their shape during embryonic development. Many developmental defects are actually a result of dysregulations in the collective cell movements that underlie morphogenesis. And that brings us to our lab's model for collective cell movements, which is Drosophila embryonic epidermal wound healing. So Drosophila, or fruit flies, are our model organism, and what we do is we take the fruit fly embryo and we use a laser to create a wound in the epidermis, and we watch as that wound is healed by the cells around it in a process that results in actually no scarring whatsoever. Now, as you watch this wound heal, you'll notice that the edges of the wound become really smooth, and that's really a testament to just how coordinated these cells are as they work together to close this wound. I'll also point out the green labeled molecule, which is myosin. Myosin is an important molecule in many biological processes for generating tension. And what myosin is doing here is it forms a cable around the wound outside of the cells that acts sort of like a purse string to pull that wound closed. Now, the other molecule that was labeled there is E-cadherin. E-cadherin is a key component of adherens junctions, which are an example of cell-cell adhesions. What we know is that E-cadherin localizes to these tricellular junctions during wound healing. And at the same time, it's removed from these linear junctions that lie between the tricellular junctions. And so throughout the wound healing process, we get these nice bulbs of E-cadherin at these tricellular junctions with little to no signal in between. And what we think is happening is that the E-cadherin and the cell-cell adhesions are helping to anchor that myosin cable and hold that tension. We have also shown that E-cadherin accumulation at these junctions is dependent on the tension in that myosin cable, and that if we reduce the tension in the myosin cable, the E-cadherin accumulation is impaired. What we're unsure of, however, is what cell ECM adhesions are doing in this process. In the diagram on the left, you see an integrin, and it's connected to the extracellular matrix outside of the cell. The extracellular matrix, or ECM, is the proteins that make up the surroundings around the cells. On the inside of the cell, it's connected to the actin cytoskeleton in red via this molecule talin. Talin is important for us because we use it to visualize the locations of these cell ECM adhesions. Now, what we do know through visualizing talin is that at the onset of wound closure, it's distributed evenly throughout the cells. However, during the wound healing process, it localizes to these linear junctions. And so we now have this nice picture of cell-cell adhesions localized to tricellular junctions and cell ECM adhesions localized to the linear junctions between them. So what we think is happening is that these cell ECM adhesions are helping to anchor that myosin cable along with the cell-cell anchors. And that brings us to the question that started my project this summer, which is, how are these cell-cell and cell ECM adhesions actually working together to close this wound rapidly? Before we got to do that, though, we needed to quantify E-cadherin fluorescence at tricellular junctions. In order to do that, we need to annotate the junctions, which requires manually clicking every single junction in every frame of every video that we want to analyze. And as you can imagine, that's quite a labor-intensive process. And so we set out at the beginning of the summer to create a semi-automated algorithm that can perform these annotations for us. The system that we came up with is we start with a raw image, which I'm showing on the right, with a dashed line around the wound. And we use a support vector machine to detect all junctions throughout the image. Since we only want the junctions that lie on the wound edge, we use an annotation of the wound edge to select only the classifier rectangles of interest. Within each rectangle, we then place a fiducial or point annotation at the maximum intensity where we think the junction may be. We've also implemented a step where the user can edit a single frame of the classifier rectangles, and we use that information to inform fiducial placement in all frames of the video, resulting in an improved annotation such as the one shown. Finally, we've implemented an interactive editing tool where the user can finalize their annotations to the form that they want. Overall, this semi-automated algorithm has improved the processing time to only 1/3 of what it used to be. And so with this new tool in hand, we set out to analyze some experimental data. Now, I didn't have the chance to be in lab this summer, but I did have the opportunity to work with some data that was produced by Clara Schimmer, a former master's student in the lab. And what Clara did is she wanted to inhibit integrins, and she did so by injecting embryos with this peptide RGDS. RGDS binds the integrins where they would otherwise bind the extracellular matrix, and in this way prevents cells from adhering to the ECM. What Clara was able to show, and what is seen quite nicely in these videos, is that wound closure is heavily impaired in these RGDS-injected embryos. And this showed us that cell ECM adhesions are actually important and required for this rapid wound closure response. So next, we wanted to see how cell-cell adhesion behaved with cell ECM adhesion inhibition. And we hypothesized that cell-cell adhesions would increase their strength in order to compensate for the loss of cell ECM adhesions. The first place that we looked was the linear junctions, since that's where the cell ECM adhesions are localized. And we saw that integrin inhibition did not affect E-cadherin removal from the linear junctions. However, when we looked at the tricellular junctions, we noticed that the control group was able to accumulate E-cadherin at the tricellular junctions by about 20% as expected. However, the RGDS group actually had removal of E-cadherins from these locations, which is the opposite of the strengthening that we were expecting. I'll also point out, however, that the initial intensity of the RGDS group is higher than the control group. And so what we think is happening is that there was an initial strengthening mechanism where the cell-cell adhesions attempted to compensate for loss of cell ECM adhesions in the unwounded tissue. However, somehow this mechanism is failing during the wound healing response. So what I've just shown is that we've been able to show that cell ECM adhesions are involved in this wound closure response, whereas it was previously thought that only cell-cell adhesions were taking part. We've also been able to show that cell-cell and cell ECM adhesions are cooperating to drive rapid healing. And so the question becomes, how are these two adhesion types actually communicating in order to do this? Sort of the classical way to think about this is that there is some signaling process interior to the cell that is allowing the two types of adhesions to communicate. However, this doesn't explain why we would see different results at linear junctions versus tricellular junctions, or in unwounded versus wounded tissue. And so, in order to explain our observations, we've created this model wherein cell-cell and cell ECM adhesions are linked via cable tension. In a normal embryo, you have this high-tension myosin cable, and it's linked at tricellular junctions by cell-cell adhesions and at linear junctions by cell ECM adhesions. However, if you inhibit those cell ECM adhesions, you get improper anchoring of that cable, leading to a lower tension. As we've already established, a lower tension results in impaired E-cadherin accumulation at tricellular junctions, which agrees with our observations. This model is able to explain that we don't see impaired E-cadherin behavior at linear junctions since it's not involved in the anchoring process here. However, the model relies on the idea that cell ECM adhesions are in fact involved in this anchoring process, which is something that we've yet to show. And so moving forward, we'd like to confirm this by performing cable cutting experiments, where we cut the myosin cable in order to measure the tension by observing the recoil. We would hypothesize that if indeed integrins are involved in anchoring the cable, then if we inhibit integrins and cut the cable, we should see a lower tension, which could be observed with a lower recoil. As well, we'd like to use talin mutants where talin has been genetically deactivated to confirm the results of our RGDS injection experiments. So hopefully I've convinced you today that we've been able to demonstrate the importance of cell ECM adhesions in wound healing, and not only that, but we've also been able to show a link between cell-cell and cell ECM adhesions that was previously unthought of. Now, sort of on a personal interest note, this coming year I'm going to be doing my PEY in engineering new cell culture techniques for microfluidics and 3D bioprinting. And I'm excited to take this idea of an external tension tuning adhesion properties and apply that in a more engineering sense to these cell culture techniques. And with that, I'd like to thank everyone who's contributed to this project, specifically Katie Rothenberg, who is the postdoc who supervised me and who's been a huge help in guiding this project along, as well as all of my lab mates for their feedback on the presentation and the project, and all of the contributors who made this project possible through their funding. And with that, I'd be happy to answer any questions that there may be.
Thank you, Raymond. Could the judges please unmute themselves and ask their questions?
Hi, Raymond. Hi, Raymond. Thank you for the presentation. That was really cool that you were able to do a lot of that image analysis during this time. I just had a quick question about, so you talked about ECAD and integrins and all these interactions, but are there any other cell-cell or cell matrix interactions that you might think could be involved, or is it really strictly just those interactions that you presented? Thanks.
Sure. So as far as cell-cell interactions, I believe E-cadherin is the one of interest because the E-cadherin removal from the linear junctions and accumulation at the tricellular junction is necessary to form that myosin cable. And so really that's the cell-cell adhesion that's really important in this system. As far as cell ECM adhesions go, this is actually sort of a new exploratory area for us because it was previously thought that cell ECM adhesions weren't involved in this process. And so integrins is the first thing that we've really explored and shown that indeed these are playing a role.